Hello, my name is Eric Lee from Curran High School. I'm in the 11th grade. I'll be talking about computational fluid dynamics of immune cells. So let's start. So let's start with some of the more questions and hypotheses. So we essentially have this theory called a secretory cleft, where when a T cell collides with another cell, it creates a little rift within the cell that tries to engulf the other cell, and we call this a secretory cleft. Now, the thing I'm studying is, do these cells essentially create these clefts naturally when they collide? And if so, how stable are these? How long do they last without active force imposed by T cells to hold these secretory clefts? And more importantly, is there any leakage? The current hypothesis is that these secretory clefts form naturally when these two cells collide, that they're relatively stable lasting around 5 seconds, and ideally they should not leak anything. All the programming was essentially done on a computer and no physical materials were used aside from a macOS running computer and an Ubuntu server. We did also test this on a Windows 10 virtual machine. The software that we used is IBAMR, which stands for Immerse Boundary Adaptive Measure Refinement. This is essentially a software that allows us to simulate fluid dynamics between cells. Visit is a software that allows us to visualize that data. Python created points and parsed out the data that came out of these simulations, and Docker allowed us to keep a consistent environment to run our code. The code repository is linked above if anyone was interested. So the essential short methodology of this project. So you would pull the image from Docker Hub to ensure a stable environment and run these Python scripts to generate points and springs that would essentially create a original simulation for a cell, try to replicate its movements. Now we put these little points into IBMR, which runs its little simulation and tries to calculate out the fluid dynamics. Um, Now, the data that comes out of this isn't readable to the human eye, so we use Visit to visualize it into images and Python to parse out those images into results. So now we would repeat steps 2 to 4 to essentially accurately simulate CD8 positive T cells and improve this over time. Now, here are some of the example initial positions that some of these T cells that we might put through generated by the Python code. Over time, these simulations became more and more accurate to how a real T cell will engulf another cell. This is currently the stage of where our simulations are at, and we essentially hope to improve upon this. So the simulations right now actually use a lot of these as synaptic connections instead of just being collided. So we need to actually remove these manual clefts that we've created and essentially just have them collide and improve upon this idea. The study is very important because we need this to essentially study leakages of different viruses that the T cell might capture, such as the ones that you might find in the HIV vaccine. Overall, this also gives a general improvement on understanding of such a new field in T cells. I hope to continue this project through 2021 to 2022 to further create these simulations and improve science as a whole. Thank you very much for listening.